Okay, thank you, Prof. Salmon. So I will ask the other speakers, Dr. Sumaya and Dr. Sulaf, uh, to join us for the question. Will not take a long time. So Dr. Sumaya with us. So I will start with Prof. Salmon. I have, thank you for your interesting talk. So you are covering the forensic toxicology and also the anti-doping. And now in Saudi Arabia, especially in King Faisal, we are planning to establish a reference lab for anti-doping. And we will start with the athlete biological passport at the beginning to be approved lab. Then we will go to the another modules, the steroidal and the growth hormone, then to full reference lab, inshallah. So back to your presentation regarding the hair sampling. So do you recommend, according to the hair sample, give the parent parent drug, not the metabolite? So can we use them and start with the hair as the screening instead to go to the urine or blood, or we should follow the order to start with urine, then the blood, then go to the hair sample?
First, congratulations for the new laboratory. It's very exciting. And can you just unmute your microphone? Sorry, otherwise I have a echo.
Okay.
Thank you. Sorry. Perfect. And yes, very good question. I would always start with urine because, urine you have high volumes and high quantities of metabolites, so the screening is easier. With hair, you have a lower quantity and very low amounts of drugs to detect. So it's much better, in my opinion, to go for confirmation in hair. And in some cases, you do not have urine and blood because they were not collected in time, because maybe the case was lately reported. So yes, maybe you have urine, but the information from urine is not useful anymore. And then in that case, of course, hair becomes the only possible to give information, to obtain information on the case.
I will go to Dr. Sumaya Al-Junaydil. So,
sorry, I see another question in the chat.
Okay, so I will go. Okay, so is hair analysis valuable to do for measuring element like zinc level to diagnose certain diseases? Okay.
Yes, the metal analysis is certainly feasible in hair. Every element can be detected and measured. Then the interpretation is challenging because there is a lack of reference values. So that's probably the most difficult part of the job in case you are trying to make a diagnosis for certain disease. But certainly you can do the analysis for zinc and all other elements or metals.
Can we use the zinc level at the hair for toxicity, not for just routine follow-up for the zinc, to measure the zinc at the hair analysis?
Yes, again, you can, but I'm not sure about the interpretation of the results. Probably with a segmental analysis, if you are able to distinguish levels in different segments of hair, one level is higher, in one segment the level is higher than the other one, then you can prove that there was an exceptional exposure to zinc, for example, or production of zinc in case of a disease.
Okay, thank you, Doctor. So my question now to Dr. Sumaya Al-Junaydil, you have a couple of questions about what is the cutoff value for the ethanol to consider the test is positive? Dr. Sumaya?
Yeah, do you hear me now?
Yes.
Yeah, do you hear me now? But again, there is an echo, please tell us. Yes. So the toxic levels, or not, we shouldn't say the toxic, the detection, or we consider it's detected level for ethanol is 10 milligram per deciliter usually. This usually correlates when the patient starts to have mild symptoms, and the lethal concentration when it's reached to 450 milligram per deciliter. Do you hear me?
Okay. Thank you, Doctor. Also, my question to Dr. Sulaf.
Do you hear me?
Yeah.
Okay.
Okay. For Dr. Sulaf Atasi, so for Raman spectroscopy, do you need to confirm your result as a portable with another method, or it's okay just to deal with them and act reaction and action according to the result?
Yeah, hello. So you always need the reference technique for Raman spectroscopy because there are many variables to consider beyond the sample. So if it's, particularly if you have a dark sample, or if the sample is in very low concentration, then you always need an analytical technique, ideally mass spectrometry, something the opposite to it.
Here also a question. The question will have a question regarding the concentration of drugs in hair follicle. It is the same or decrease with the time. So the second question: do you prefer analysis of drugs hair only in case the blood or urine not available? So, I mean, is it sensitive? This is the question for Prof. Salmon.
Yes. Thank you again. Two very good questions. The first one is indeed there might be a decrease over time. I need to be more specific. If you mean that with time after collection, no, hair is stable, so the concentration would not change after the hair is cut. But if you mean with time after the intake, yes, because time, washing, cosmetic treatments, these are all factors that affect the concentration in hair. So in this case, yes, the concentration might decrease after time, but I mean months, and we are not talking about days. Second question, it really depends on the purpose of the investigation or of the analysis. Hair is, of course, the matrix of that you can use when you need information about past use, chronic use, or in general, when the drug use has happened weeks or months before the analysis. Otherwise, if it's about a recent intake, urine and blood are, of course, much better.
Okay, so now I will conclude this interesting session. I would like to thank Dr. Sumaya Al-Junaider and Dr. Sulaf Atasi and Prof. Alberto Salmon. We will go to a break for 20 minutes and then we'll start the last session for today with the moderators, Dr. Salam Saaduddin. And thank you again for our speakers and also for all the audience. Thank you.
Thank you.
Thank you.
Bye-bye, Sulaf.
Thank you.